Hi, everyone. I welcome you all to the 2025 RNA Collaborative Seminar Series hosted by the RNA Society and co-hosted by IMB Mainz and RNA Research Center in Poznan, Poland. In this virtual series, we have some really fascinating talk on long non-coding RNAs and RNA-DNA hybrids. For audiences, if you have any questions, please mention them in the Q&A section of the Zoom interface, not in the chat box. Okay, so now I'm really pleased to introduce our first speaker, Professor Brian Luke. He has immense contribution in telomere biology and cellular senescence. His research focuses on this very fascinating long non-coding RNA, telomere repeat containing RNA or TERA, and and the role of RNA-DNA hybrids in telomeric dynamics. So I would welcome Professor Brian Luke for his interesting talk.
Okay, thanks very much, Aaquib. And thank you also to everyone who has joined. And I'm definitely glad, to be able to share some of our data with you today, where I'm going to indeed be talking about a long non-coding RNA called TERA, and how it basically affects the rate of senescence when telomeres start getting short. Right. So first thing I'd like to just remind you a little bit about telomere biology. So just reminding you that every time a cell divides, the telomeres get shorter and shorter and shorter due to the end replication problem. And eventually you get these critically short telomeres that activate the DNA damage checkpoint. And what that does is it causes the cells to arrest in a permanent manner. And we call this permanent cell cycle arrest senescence. Right, so when it's associated with telomere shortening, we specifically refer to it as replicative senescence. And I just want to remind you that these senescent cells, as they accumulate, the more cell divisions we have, we now know that they directly contribute to aging. Right, so the rate of telomere shortening then really needs to be tightly regulated. Otherwise, this can lead to telomere biology disorders. So what happens is if the telomeres shorten too quickly, then what happens is that at a young age, you end up with too many senescent cells, and this drives aging-like phenotypes even at an early age. At the same time, you want telomeres to shorten. Telomeres should shorten because this acts as a tumor suppressor, right? So if a cell starts to divide in an uncontrolled manner, basically it will eventually stop because the telomeres get too short, right? So telomere shortening, it really needs to be balanced. You don't want it to happen too fast, but you want it to happen. And what I'm going to tell you about today is how this balance of the right amount of shortening is achieved through a long non-coding RNA that we have named in the past TERA, which stands for telomere repeat containing RNA. Right. So a little bit about the TERA RNA. So it's an RNA Pol II transcript, and the initiation of transcription actually starts in the subtelomeric region and goes into the repetitive region of the telomere. TERA molecules can be detected from every chromosome N, so all telomeres transcribe a TERA molecule. It's conserved from yeast through to humans. The data I'm going to be showing you today is, in fact, from yeast experiments. And what we've shown in our lab, and other labs have now shown this as well, is that TERA readily forms RNA-DNA hybrids or R-loops. So these three-stranded structures where you get an RNA-DNA hybrid and one strand of DNA is displaced within the double helix. And what our lab has been trying to figure out over the past 15, 16 years is really, is TERA physiologically relevant and what is it doing? Right. So the question we set out to ask is, do we want Terra or not? And the answer I can tell you is yes, but just a little bit. And I'm going to explain that in that talk. So the first part of the talk, I'm going to tell you some published data about how Terra forms R loops at short telomeres, specifically at short telomeres, to drive repair and prevent senescence. Then what I'm going to tell you in the second part is that Terra R loops must be removed at short telomeres to drive repair and prevent early senescence. And this is a story that is currently in press. So the long and short of it is Terra at short telomeres is good. And then I'm going to tell you getting rid of Terra at short telomeres is good. So how do these two things make sense, even though they're opposites? Okay. So the first thing we set out to do when we were trying to figure out what is Terra doing is we created a single telomere that doesn't have Terra anymore. And we did this on chromosome 7, the left arm, so 7L, and we put a star there because it's 7L star, it doesn't make Terra. We could confirm by qRT-PCR that indeed all the other telomeres that we checked, we could detect Terra molecules, but not at this mutated telomere that we made, right? We could not detect a single molecule of Terra. And we wondered, of course, what was the effect of not making Terra on the telomere? We tested many different things. We tested the chromatin structure, we tested telomere length, and there weren't any differences. But we did notice a difference when we lost telomerase and we started going through this senescence process. And what we noticed is the following. If you look at a normal telomere that makes Terra, so let's look at telomere 1L, what you see is that the telomeres shorten with every population doubling, PD, and then when they get very short, the shortening rate tapers off. Okay, and we can see that every single time we do this experiment, when telomeres get short, the shortening rate changes. And at the Terra-less telomere, this doesn't happen. The telomeres shorten and they just continue to shorten at the same rate. Now, it had previously been shown in the field that this change in shortening rate is due to recombination. So short telomeres, they actually start to recombine. And this maintains their length for a little bit and delays senescence. So we thought that if this difference is due to homologous recombination, then if we eliminate recombination, we should eliminate this difference. So we performed the same experiment in a strain that cannot do recombination, and you could see that indeed we could completely eliminate this difference. So this gave us our first indication that Terra at short telomeres may be driving recombination and lengthening. So if that's true, then we wondered then if Terra is regulated in a length-dependent manner. Is it only there or mainly there when telomeres get short? Because this is where it seems to be needed. So we did this many different ways to address this, but I'll show you one of the ways that we did this. And one of the ways we did this is we made one short telomere in a culture. So we took a culture of yeast that had a telomere that was genetically manipulated with FRT sites, and we could basically flip out a big circle out of this telomere and leave ourselves with a single telomere. And this was very convenient because then we knew in a culture of yeast, we knew exactly which telomere was the shortest telomere. And then we asked what happens to Terra at that telomere. And what we saw is that compared to the control strain, when we make a short telomere, what we call VST, very short telomere, we get a 3 to 4-fold increase in Terra levels. But if we look at other telomeres, we don't see any change in Terra levels. So Terra seems to specifically get upregulated at short telomeres. And what about R loops? So we also showed that, I won't show you the data, we showed that R loops and RNA-DNA hybrids also accumulate at short telomeres. Do the R loops drive recombination at these short telomeres? So we did a similar experiment. We used this system where we could make a short telomere, and what you can see is that we looked at recombination factors via chromatin immunoprecipitation. And what you can see is that when we make a short telomere, we accumulate Rad51 at that short telomere. But when we overexpress RNase H1, the enzyme that degrades RNA-DNA hybrids, we lose Rad51 at that short telomere. So the RNA-DNA hybrids are important to accumulate recombination factors at the short telomeres. And you can see at other telomeres that we didn't shorten, we don't see any Rad51 at those telomeres. Okay, so the model that we put forward at the time is the following, and this model still holds true, we've just added to it, is the following. When telomeres are long or normal length, what we know is that Tera is transcribed by RNA pol 2, but the RNA-DNA hybrid is quickly removed by RNase H2. And it removes it before the replosome passes by to allow smooth replicating of the telomere. When telomeres shorten, we know that RNase H2 is lost at short telomeres. So now when Tera makes an R loop, it's no longer removed. And what we believe is happening, and we have some evidence for this, is that the replication machinery now encounters the RNA-DNA hybrid. This causes replication stress, and this then drives recombination. So this causes the recruitment of Rad51, and then you get recombination with other telomeres, and this maintains the length of the telomere. Okay, so this was what we believed in 2017 when we published these results. We can also test this model. So one of the things then you can test here is that, okay, if it's true that you need Tera R loops to accumulate in order to get recombination, if you prevent R loops, then you shouldn't get recombination anymore, and the cells should senesce very, very fast. And we do that by overexpressing RNase H1. Another prediction is that if we eliminate RNase H2, then we should get recombination all the time at telomeres, whether they're long or short, and we should delay senescence. And I can tell you that both of these are true. So if we overexpress RNase H1, we can see that the loss of viability with time is very, very quick. So they senesce very, very fast when there are no hybrids. And if we take away RNase H2, now we have more hybrids, we delay senescence, and they senesce very, very slowly. Okay, so this was the model that I just talked to you about. So to summarize, Tera forms R loops at short telomeres to drive repair and prevent early onset senescence. And as I said, this data is published. Now, in the meantime, we published another paper in 2023, and what we showed is that RNA-DNA hybrids prevent resection at dysfunctional telomeres. Now, this is very important because in order to get repair by recombination, you have to get resection to expose single-stranded DNA to allow Rad51 loading. So this is a bit of a conundrum because what I just told you is that Tera RNA-DNA hybrids promote homologous recombination. But what I'm also telling you is we found that Tera RNA-DNA hybrids prevent this important step here, and that is resecting the 5' end. So how can accumulation promote HDR and at the same time inhibit an important step at HDR? So we hypothesized that there must be another step here that once the hybrid accumulates, that removes it again, right? So Tera R loops must be removed at short telomeres to drive repair and prevent early senescence. And yeah, this is current, as I said, this is currently in press now. So we got evidence that this is true with the following experiment. So again, we looked at rates of senescence, and you can see this is normal senescence in the black curve. If we overexpress RNase H1 and we remove hybrids, we get fast senescence. I already told you that. But what we also found is if you overexpress catalytically dead RNase H1, what we know is that this stabilizes hybrids at the telomere. This also causes fast senescence. So no hybrids or hyperstabilizing hybrids, they both drive rapid senescence, and they do so in a recombination dependent manner. If we do the same experiment in a RAD52 deletion, we don't accelerate senescence any further, right? So it's going through recombination. Okay, so what enzymes are removing hybrids in senescent cells? We had three main candidates. In Saccharomyces cerevisiae, there are three main what we call R-loop or RNA-DNA hybrid resolvases. There's RNase H2, what I talked about, RNase H1, which I've also talked about, and SEN1, which is the homolog of senataxin. So we're pretty sure that it's not RNase H2, because what I told you is that when telomeres get short, RNase H2 is actually removed from short telomeres. It's not there anymore. So what we predicted then is that then you get this replication stress at the stabilized hybrid, but then one of these two factors, H1 or SEN1, must come in at this step and remove the hybrid so that then you can get resection and repair. So we wanted to test this model. Do H1 and SEN1 get recruited to short telomeres? So the experiment we did is we took cells through senescence with epitope tagged versions of RNase H1, RNase H2, and SEN1. And we took time points at early and late senescence, and we did CHIP at telomeres. And what we found is, as we have already published in the past, is that when telomeres start to get short, we lose RNase H2 at short telomeres. If we go even further down this curve, we lose even more. Okay, so H2 is lost at short telomeres and hybrids accumulate. But as RNase H2 is lost at short telomeres, what we now find is that RNase H1 accumulates. So if you compare early senescence to late senescence, you can see that H1 increases 2.5-fold at the telomeres, no matter what telomere you're looking at, 1L or 15L. SEN1 accumulates a little bit at short telomeres, but not so much. It seems to be a little bit more there than early senescence, but one has to also remember SEN1 travels with the replisome, so it would be there anyway, and it seems that maybe it has a little bit longer dwell time at short telomeres. We haven't looked into that much further. And what we also found is that if you actually look at R-loop levels at telomeres compared to wild type, you have to deplete all three resolvases, SEN1, RNH1, and RNase H2, before you get a significant increase in R-loops. So they all work together, all three, to metabolize RNA-DNA hybrids or remove RNA-DNA hybrids at short telomeres. And here are all the other different combinations. So all three have to be gone. Okay, so then the next prediction was, okay, if RNase H1 or SEN1 don't get recruited, then what we would expect is that this RNA-DNA hybrid would continue to prevent resection, and we should not get RAD51 loading. And this is the experiment that we did. So what we did is we took telomerase mutant cells, and what you can see, and again, we checked in gray early and in orange late senescence, and what you can see is that indeed RAD51 accumulates as cells go late into senescence. So at short telomeres, RAD51 accumulates. Indeed, what we found is that when you delete RNase H1, RAD51 does not accumulate at the short telomeres anymore. So you really need RAD51 at this, RNase H1 at this step to remove the hybrid so that it doesn't prevent resection and you can get proper RAD51 loading. And the final piece of data that I'd like to show you is that, does this have an effect on rates of senescence? And it does. So I just want to remind you, so there are a lot of lines here, so I'll tell you which ones to focus on. If you look at the black line, this is normal senescence. Now I told you if you delete RNase H2, you get delayed senescence. That's the orange line. So you get delayed senescence. But now if you delete RNase H1 on top of that, you reverse this. Not fully, but you reverse it to an extent. And that's again because you no longer get Rad51 loading. The same is true for SEN1. Again, the black line is normal senescence. RNase H2, you get delayed senescence. And now if we make a mutant of SEN1, we revert this back. So you need SEN1 and H1 in order to, in order to get the right proper rate of senescence. Right. Okay, so putting it all together one more time, again, at a normal telomere, Tera gets transcribed, it forms an RNA-DNA hybrid, RNase H2 removes it before the replosome comes. Then we know that when the RNA-DNA hybrid is stabilized, this activates the checkpoint. And in data I haven't shown you, what we know is that the checkpoint is required to promote RNase H1 to go to the telomere and remove this RNA-DNA hybrid. This then allows resection, Rad51 loading, and regulates the rate of senescence. So in short, this is how the cell regulates the rate of senescence so carefully. It's by an RNA-DNA hybrid. And what's cool is that it's really only the short telomeres that get activated to do recombination. And that's how you get this nice tight balance. So I just want to especially point out Fabio Bento and Matteo Longoretti. So this guy and this guy who did the majority of the work that I've shown to get. And again, thanks for joining. And if you have any questions, I'll do my best to try to answer them.
Thank you, Professor Luke, for a really interesting talk. If you have any questions, please put them in the Q&A section. Okay, for now there are no questions. So, Bran, I found it really interesting. I was curious about Tera biogenesis. So, does Tera length or its composition changes at different length of telomeres?
Yeah, that's a good question. And the answer is no, it doesn't. So it doesn't matter if you have very long telomeres or very short telomeres. It seems that transcription of Tera only goes a certain distance into the repetitive region and then it stops. So remember, there is no transcription termination site, right? These are repetitive. So it sort of always randomly stops. And what has been shown is that it goes about 150 or 200 base pairs into the telomeric repeat and then stops.
Okay.
Yeah.
Okay. One more question. Okay, I think we have a question. Yeah, so there's one question. The question is, perturbing RNH1, does it also affect angiogenin substrates?
Oof. I can't answer that. I have no clue. I'm sorry. I don't know the answer to that question. Yeah.
Okay.
Can I answer a question from here? Sorry, because I cannot use the Q&A.
Sure.
Yeah, sure.
So I was wondering because this RNA seems to work mainly in cis mode.
Yep.
But there is some evidence that it can also act on neighboring telomeres.
Right.
It has some activity like binding proteins, and I think I'm not sure, but I think it also can modulate the chromatin environment to keep the heterochromatin. So basically, this version of packed chromatin. So do you know what is the reason of that, and how your experiments actually explore this more trans functionality?
Right. So the trans functionality of Tera is what I think has never been shown is that Tera can act, that in vivo, under normal conditions, Tera can act in trans. So what has been shown very clearly from the Ligner lab and from the Azzalin lab is that if you express Tera off of a plasmid, or you introduce, for example, RNA oligonucleotides, then we know, so if you force the system, you can indeed force Tera to go to telomeres, to whatever telomere you want, in fact. But the question, the thing is, does this actually really happen? You know, if you have a short telomere, does the tera produced at that telomere then go to other telomeres? And there has been no evidence. We have not seen any evidence of this. So if we make a short telomere, we don't see tera upregulation at any telomeres, nor do we see RNA-DNA hybrids at any other telomeres. But it is cool to think that you can push the system though, and you can put an RNA-DNA hybrid. And this may have important implications, for example, telomere biology disorders. Right, that you can then drive them to recombine.
Okay, yeah, that's really cool and complex model.
Yeah, thanks.
Thank you.
Oh, Aaquib, you are muted.
Oh, yeah, sorry. Yeah, so there's one more question from Onofrio. The question is, what might inhibit the activity of RNase H2 to shorten telomeres?
Yeah, so there's nothing inhibiting its activity. It is no longer getting recruited. So the thing is, RNase H2, what we've shown is that it interacts with telomere binding proteins. So it's just a matter of numbers. As the telomeres get shorter, you have less of these telomere binding proteins and less recruitment of RNase H2. So it's really just the fact that you have less telomere, you have less, it's basically RAP1 and RIF2 that bind it, and then this no longer then is enough to recruit RNase H2. But activity is not affected as far as we know.
Okay, all right. I think we have time for another question. Ren asks that is there analogous mechanism at telomeres in mammalian cells?
Yes. So I can just, it's a very, very simple answer. Yes. As telomeres get shorter, you get more tera at short telomeres, whether it's yeast, whether it's human cells, and you also get more RNA-DNA hybrids. And RNA-DNA hybrids at telomeres promote recombination at the telomeres in human cells, just as they do in yeast.
Okay, we don't have any more questions. So thank you so much, Professor Rook, for the very interesting talk and answering all the questions. That wraps up this session of the talk. Now over to Dr. Warkocki.
Thank you.
Yes, thank you. I'm taking the relay. Welcome everyone to the second part of today's RNA collaborative seminar series. It's time for the second talk. Our speaker is already on. So our speaker is Barbara. And now listen carefully. Uszczyńska-Ratajczak. That's the full name. So you have plenty of time to get acquainted with that. Very shortly about Barbara, she has studied in Wrocław, Poland, and then later at Adam Mickiewicz University in Poznań, Poland, and with a specialization in bioinformatics. And then later she completed her PhD at the institute where she currently works, Institute of Bioorganic Chemistry, Polish Academy of Sciences in Poznań. And after that, she moved to Barcelona, just not to say something stupid, Center for Genomic Regulation, Barcelona, and Rodrigo Guigo was her PI at the time. She got involved into the Gene Code project, a very well-recognized project of annotating human and other vertebrate genomes. And she then discovered her love and engagement for long non-coding RNAs and became part of an ENCODE project to identify those in the genomes of different vertebrates, and I think with a strong links to zebrafish. Zebrafish was her organism. And I believe that, this is what she will be talking about. Just to briefly tell you, I think one more thing, Barbara is a very successful scientist in Poland and our institute. She has funding from National Science Center and other agencies. And I think worth mentioning is that she has been awarded a L'Oreal prize Poland for Women in Science some time ago. I think that's also an important part in her career. No more introduction from my side. Barbara, floor is yours. We are listening carefully now.
Thank you so much. Thank you for this nice introduction. Thank you for having me here. Actually, I decided to talk more about human and mouse instead of zebrafish because we explored zebrafish topic recently and also yesterday during my PhD student defense. So it's actually a very nice example with Terra because what I'm mainly doing is building annotations. So people who are working with specific low non-coding RNAs, they have a good foundation for investigating their function. And in contrast to previous expectations, 80% of the human genome is transcribed, but only 1% can produce protein coding genes, and this actually actively transcribed genome is a good source for various transcriptional various regulatory units, including low non-coding RNAs. It was really, I think, after when the Human Genome Project was accomplished, it came with a very big surprise that our genome only contains 19,000 of protein coding genes, and this tiny number, actually this tiny fraction, is outnumbered by low non-coding RNA genes that are estimated to between like mid to high thousand. I think 100,000 is the latest estimation. So what we mainly do is we look at the sequence of the human genome in mouse and zebrafish genomes, and because this information itself doesn't provide any information about functional elements and those elements that are important for our body. So what we do, we try to add meaning to it by actually defining precise location of transcription start site, location of exons, introns, how they are connected. And here's the example how we annotate things. This is Umlilo, very famous low non-coding RNA for immune prime cells in mouse. So what GenCode is actually evidence. What GenCode project does, we produce evidence-based, manually curated gene annotation for human and mouse genomes, and we also experimentally validate this annotation and produce high-quality models. And those models are used by big consortia projects such as ENCODE and GTEx. And for this reason, GenCode became a gold standard for low non-coding RNA annotation. And actually, GenCode has been annotating genes since 15 years, and you can see that protein coding genes were more or less stable during this time, while low non-coding RNAs, the number of low non-coding RNA genes steadily increased, and at some point it actually started exceeding the number of protein coding genes, and this is the state for human, but for mouse this is less optimistic, so we still see this this discrepancy. So of course the main difference is that low non-coding, the final product of low non-coding RNA gene expression is RNA, and it has several implications. And because many people still consider that low non-coding RNAs are not really are non-functional element of the genome, and there is like a hot debate about their functionality in general. So and this also comes from the fact that low non-coding RNAs, they break this golden rule of protein coding genes that one gene equals to one function, and they operate via multiple modalities. So some low non-coding RNAs, they act simply via their act of transcription, and they are regulating the neighboring gene. Some of them can encode short peptides, as there is no clear difference between low non-coding RNAs and protein coding genes, so evolution can actually clear those barriers. And then they are the most fascinating element, the most fascinating group, acting like in a sequence dependent manner. Of course, there are some non-functional ones, but data seems to suggest that the vast majority of them are actually operating like or are contributing to organismal fitness via their mature molecules. So this brings also another element of complexity, because long-non-coding RNAs, they have modular architecture with short, like with their functions encoded by discrete domains that help them to interact with other molecules like DNA, RNA, and also proteins. But what is important that not entire function is not entire sequence is needed for their function, which is actually hampering the detection of conservation of long-non-coding RNA molecules, and also these are the molecules that are known to be rapidly evolving in contrast to protein coding genes. But what is very important is that alternative splicing shapes functional potential of long-non-coding RNAs by including or excluding exons with different functional potential. So, I mean, the big technological revolution in the genomic world, including also development of different sequencing platforms, pushed annotation of long-non-coding RNAs far ahead. But most of the methods, they are still based on short read sequencing. And using this analogy from Hagen-Tilner's lab, where we go from individual words to complete phrases, which is now enabled with the long read sequencing. So we have all the elements, but then the long read sequencing helps to put them in specific context. And this context is really important, because then depending on the context, the complete phrase can be different. And this is also what happens with the function. So because there are no, I mean, as you saw, probably, as you noticed probably from the slides, I mean, there is no good proxy for function for long-non-coding RNAs, because we cannot depend on conservation. It's really difficult to detect functional elements. So basically, still, long-non-coding RNA annotation is simply based on the detection of transcriptomic evidence. And since long-non-coding RNAs are lowly expressed compared to protein coding genes, we use this technique of targeted RNA sequencing, which we called capture long read sequencing. And this method is based on RNA probes. Actually, now we are using RNA probes. So oligonucleotide probes that are complementary to exons of annotated long-non-coding RNAs, or regions where we suspect to find novel molecules. And those oligonucleotide probes help to boost the presence of long-non-coding RNA molecules in the sequencing library. And then we use long reads to build the models and to maintain this connectivity that is important for their functional characterization. So CLS can actually address two challenges for long-non-coding RNA annotation. So first is to complete existing gene structures and quite often detect novel, previously unknown functional potential, and also extend the annotations in suspected regions, and try to find some long-non-coding RNAs that are like biologically relevant as well. So one thing that we also noticed that the number of long-non-coding RNAs that we were annotating with CLS at some point reached plateau. So we noticed that we are, with the experimental design that we were using, we were better at annotating novel transcripts for known genes rather than extending the annotation. So what we did for the phase 2 of CLS project, we actually focused mainly on the regions that are outside GenCode annotation. So we wanted to actually expand the GenCode catalog as much as possible. So we targeted different regions, including also different long-non-coding RNA catalogs that are in Navy, and some enhancers, and also short RNA molecules to look for the potential host, to look for their potential hosts. So we also noticed that to maintain, like to boost this connectivity, we should concentrate on sequencing, on like identifying full-length molecules. And we actually, Sylvia Carbonell-Sala from Rodrigue's lab, where I did my postdoc, she developed a new technique that helps to actually boost the presence of full-length molecules. And this method is using two rounds of full-length molecule enrichment. First this is like cap trapping, so this biotinylation that helps to get capped molecules, and then those adapter ligations which help to get the full-length molecule. Important thing is that this technique is mainly designed for polyadenylated non-encoding RNAs. So actually, when we compared cap trap with different standard library preparation methods and different platforms, we noticed that it has actually quite nice performance with, I mean, depending on the platform, but it can reach up to 80–90% of full-length molecules. So having this increased connectivity, we combined CapTrap with the CLS method, creating a new modality. So now we focus on non-encoding RNAs, which are also full-length. We to help to actually increase the quality of GenCode catalog, and also to kind of motivate, like induce better functional characterization. So we also, I think it's very important to look at the non-encoding RNAs in specific context, like in matched context, where that's why we used a set of tissues and samples that were matched between human and mouse, and we were also able to explore adult and embryonic stages. So we noticed that actually capture works, because the green ones are the samples sequenced with unbiased sequencing, and then the blue ones shows the enrichment of our non-encoding RNAs. So, I mean, of course, it varies across samples, but we notice a clear enrichment. So one thing that we did is to analyze this huge data set. So we were concentrating on the full-length transcript models that were supported by cage tags, which are the gold standard, phantom cage tags, which are the gold standard for accurate annotation of transcription start site. And to make sure we have a complete 3' ends, we were looking at the poly A tails detected directly in the reads. So we are using standard merging, where simply based on the intron chain, we were collapsing aligned reads into a non-redundant set of transcript models. But of course, we noticed that knowing the tissue, like knowing the sample origin, is also very important. But we wanted to further, because this was like massive set, we wanted to further remove the redundancy, and we merge transcript models across samples, but this time we kept their boundaries. So we used this anchored merging strategy, where first we anchored support ends. So if the transcript was full length, we didn't merge it, and this is a good example that you have at the bottom. So this shorter transcript that has exactly identical intron chain was maintained. So we noticed that using intron chain as a basic unit was actually helpful, and it helped to it helped us to reduce the size of the set. So now when we look at the classification, we have the sample. So we know that there are some pre-capture specific transcripts. There are also some that are detected by both methods, but the majority actually comes from the pre-capture. So known, these are known transcripts. Novel, these are novel transcripts for known genes, and intergenic, these are transcripts that are likely to generate new non-encoding RNA genes. And we observed similar performance. Actually, for mouse, it was much better, I think, because mouse was lagging behind human. So we were actually expecting to see some improved performance. One thing that is important, we wanted to look at the completeness of transcript models. So we were able to annotate more than 80,000 of novel CLS models. And we look at the completeness of their fine prime ends with the cage data. So this is phantom cage that is dependent on the expression. This is PROCAP data that is coming from machine learning. So these are machine learning generated tags, AI actually generated tags. And what is important is that the combination of the support for novel CLS models was more or less around 36%. And this was slightly lower compared to already annotated non-encoding RNAs from the set that we targeted, and lower compared to protein coding genes. But basically this difference was driven by cage data that is expression dependent. So since majority of those models were coming from the post-capture data, it is actually quite expected that we won't be able to see support for them in the unbiased data. So I think there is a huge potential for AI-based solutions in genomics. Another thing that is quite well, we actually look at the ENCODE data to validate generated models. So ENCODE consortium has elements that they called candidate cis regulatory elements, CCREs. And I think currently there are like 2.3 million of those units. And we use this data set to validate the long-encoding RNA predictions, like to make sure that the long-encoding RNAs that we generate are actually genuine. And we observed that 89% of novel CLS models are supported by those CCREs, so the histone modification associated CCREs. And this support is comparable to long-encoding RNAs and protein coding, like annotated long-encoding RNAs and protein coding genes. And it's slightly lower compared to controls. One thing that is surprising is that those promoter-like elements and enhancer-like elements, P stays for proximal and D stays for distal. So for novel models, we are actually mainly obtaining support from distal models. And this is how, because if there is no evidence of functionality, elements are being labeled by the proximity to the known transcription start site. So we see that they actually correspond to distal, but since we annotated things, now this induced change in the ENCODE set, and about 8% of distal enhancer-like sequences were now converted to proximal, which reduced their overall proportion from 60, like almost 63% to 56. Many CapTrap CLS models are actually tissue-specific, which actually shows that, I mean, that this is what we expect from long-encoding RNAs, but then it also shows that including like quite a big range of samples in those experiments is also very important. So then the CLS set was actually combined with the GenCode set. And only 30% of the data, more or less 30% for human and 30% of mouse, were incorporated into GenCode. And this boasts a huge increase in the total number of non-encoding RNA genes, far exceeding what we know far exceeding the number of protein coding genes. And for the first time, actually, we have the number of non-encoding RNAs for mouse is higher than for human, and the same for transcripts. So one thing that is also important is like there are different long-encoding RNA catalogs, and those catalogs were created using different methods, and because of that, the overlap between annotation is really poor. So one of the goals was to create a unified long-encoding RNA. So basically target all those catalogs, improve their annotation, and create something something meaningful. So basically, we want to consolidate annotations and then reduce the problem with downstream functional analysis. And targets from non-coding RNA annotations were actually the most productive ones. So we see the blue ones are novel genes coming from specifically non-coding RNAs, are they targeted or not? And we noticed that when we look at the performance of annotations in terms of novel loci and the cage support that we are getting, we were able to expand the GenCode 27 catalog, going to 47, because this is the GenCode set that contained all the non-coding RNAs annotated, and the green part is showing CLS transcripts that are still pending to be incorporated. So the overlap between annotations and the merge set was actually is much higher for GenCode 47. But there is still room for the improvement. So we try to unify things, but still long way to go. I think this is related mainly with the limited set of tissues that we targeted. And also, I mean, targeting the same region is one thing, but also expanding the number of isoforms is another thing. So here's the example of non-code annotation, human CapTrap CLS models, and final GenCode annotation, which is much more comprehensive to the initial one. What we also wanted to look at is the conservation of non-coding RNAs, because from zebrafish to human, different genomes harbor actually similar number of protein coding genes, but what they vary is what the difference makes the proportion of non-coding genome. So taking into account all the elements that we were discussing before, it was really hard to find a good method to track non-coding RNAs that are potentially functional in the sequence-independent manner. But we concentrate on this positional conservation, which is actually conservation of genomic neighborhood and keeping the relative transcriptional orientation. So that helps to actually achieve this goal and look at the positional conservation without information about sequence. So what we do actually is we concentrate on searching for blocks, like conserved genomic blocks, and this is what we did in collaboration with Rory Johnson. And we designed connector, and connector works in two modes, exonic and genic. And it helps to overlap it helps to overcome this sequence and structural divergence between conserved non-coding RNAs between different genomes. We also know from our initial analysis is that the Synteny-based tools are not really comfortable with lifting longer sequences. So it's much better to lift shorter ones. That's why exonic mode is the default mode for connector. It also works with different categories. One thing that is very important from this, that connector uses this reciprocal approach. So to call an ortholog, it will look for this direction from both ways, human to mouse. Actually, with the expanded annotations, we were able to expand also the catalog of non-coding RNAs orthologs, and many of them seems to have a disease-related function, which, of course, this is only a prediction, but this shows that improving the annotation, improving the genome assemblies also helps to get better models, animal models for investigating human-related diseases. And this is the example of Torling, which is disease-associated oncogenic non-coding RNAs, and those regions highlighted in gray shows the synthetic regions between human and mouse. So, with the summary, I think it's very important to work with complete annotations, and the annotations have huge impact on the downstream analysis. We were able to apply CLS method to largely expand long-encoding RNA annotations. I think this is the biggest increase since the Human Genome Project. So now what is happening is we are trying to revise those annotations. And these are my acknowledgements, and this is my lab. And with that, I would like to thank you for your attention. And if you have some questions, I'm happy to take them.
Thank you, Barbara, for the interesting and comprehensive talk. And we do have 2 questions, or even 3 of them right now, and we are waiting for more. So I will start with reading them in the order of appearance. Question 1 from anonymous person. On average, do you find big deviations from previous annotations? For example, many more exons.
We do, and this depends on the context. And I think what is really becoming the problem now are the read-through transcripts, because we also see that something that we previously considered to be 2 different loci are now becoming 1. So I think this has to do, I mean, the support of external genomic data is needed, because the fact that we see transcriptomic evidence, it doesn't mean that it will act as a single locus on the functional level. And one of them is actually Chaser, the famous non-encoding RNA from Igor Ulicki, that has this regulatory mode that it can first have the text as 2 separate genes, and then there is this read-through transcript between non-encoding RNA and protein encoding gene.
All right. Then the second question. Approximate percentage of polyadenylation signal consensus AAU, AAA in newly annotated. I assume, what is the percentage?
I don't remember the percentage. I remember because we do not look at the canonical polyadenylation signal. We are looking at the long-encoding RNA at the long-encoding RNA data, so long read data, and we are getting those stretches of A's within the read. And if there is a read, if there is this poly A stretch, we will call the read full length. But then for the long-encoding RNA to be annotated within GenCode, it has to have the support of this polyadenylation signal. And I would say 100% of long-encoding RNAs annotated by GenCode are polyadenylated, because GenCode doesn't want to annotate non-polyadenylated ones so far.
Okay, so that's very, very accurate and definitive answer. Okay, now Lydia Dykes asks, will the unified long-encoding RNA catalog contain information on RNA structure and function?
Unified long-encoding RNA catalog contains models annotated by different resources that are now fixed and incorporated into GenCode. But this is a good starting point for the functional validation. So there would be information about the sample origin, I guess. So I think there is or if not, there would be a tag supporting the sample it is coming from, whether this is only expressed in 1 tissue or has a multiple tissue origin. But GenCode doesn't provide information about RNA structure and function. This is purely catalog of genes, but this information can be gained from other resources.
Okay, and now the last for now question, Jairam Murphy. Correct me if I'm wrong, but or maybe I'm sorry if I'm wrong. Is CapTrap suitable for non-coding RNA without caps? And how would you differentiate from non-coding RNAs and mRNAs in CapTrap?
I mean, there is no other way of doing this only by looking at the support. But of course, this is suboptimal, because the fact that we do not, and this is related to the connectivity. The fact that we have a cage support, it doesn't really mean that this RNA is fully totally complete. It means that this is simply supported by the tag. But the cap trap was designed specifically to look at the capped London coding RNAs. And the only difference that we could see is to look at the elements with cap or without cap. We noticed that cap trap is actually is not 100% effective because some spikings that are not capped, we were also able to look at them. I mean, to gain them with cap trap, but it was like below 3 or 4%. So this is still very efficient. But I think there is this specific protocol. I think it was published in Nature Communications for capping, for investigating non-capped London coding RNAs. So I think I would, if you are interested in non-capped London coding RNAs, I would look at this one. NAPCAP, yeah, sorry, NAPSEC is the method that you are looking for.
Okay. So Barbara, thank you again for the talk. Thank you for answering all the questions. Many thanks to all the attendees. And I got an information what will happen in exactly 1 month, on June 11th. There will be next RNA collaborative seminar series meeting, and it will be hosted by the Yale Center for RNA Science and Medicine and Harvard Medical School Initiative for RNA Medicine. And so I hope we meet all in 1 month. Thank you guys all.